The third form of structures that we can see within proteins is the tertiary structure. We know that, first of all, there's primary structures, secondary, and now we are on to tertiary structures. And in tertiary structures, we see the interactions between various R groups of different amino acids. Remember that the primary structure is simply the amino acid sequence. The secondary structure is hydrogen bonding, but not with the R groups, but with the backbone of the peptide chain. Now, here in tertiary structures, we actually see interactions between the R groups. That is the distinctive feature between tertiary and secondary. In tertiary structures, we see R groups interacting. Now we're going to go through how we get tertiary structures and their stabilizing features. So a tertiary structure, we have our hydrophobic groups sticking on the inside, and we have our hydrophilic groups on the outside. This is the main driving force of tertiary structures. So essentially, when we have our tertiary protein structures in aqueous environments, it is not favorable for those hydrophobic nonpolar groups to be interacting with our hydrophilic polar environment. That is why the hydrophobic groups will aggregate in the center and interact through London dispersion forces or van der Waals interactions and avoid that aqueous environment. And we will see that the polar groups will actually aggregate on the outside and interact with that aqueous environment because that is favorable. So the first driving force we can say for tertiary structures is the hydrophobic effect and the R groups clustering on the inside to increase the entropy and decreasing the energy required. Now, tertiary structures can have alpha helixes, as we can see over here. We can have beta-plated sheets. We can also have disulfide bonds. Now, disulfide bonds are actually two cysteines that are deprotonated. So cysteine has a pKa of 8.3. So whenever the pH is above 8.3, it'll be the cysteine will be deprotonated, and the two cysteines can interact and form a disulfide bond. Di means two, sulfide simply refers to our two sulfurs. Now, in our tertiary structures, we can have these disulfide bonds. Now, we can imagine that if our pH of our system decreases below 8.3 and the sulfurs become protonated, we will no longer have this disulfide bond. Okay? So for tertiary structures, it's important that the pH of the system remains above the pKa of cysteine in order for us to see disulfide bonds. Now, a third feature, which is very intuitive, is the electrostatic repulsions or attractions between our R groups of our amino acids. So in our tertiary structure, we now have the capability of our R groups to interact. Now let's say we have a negatively charged R group like glutamate, which has the carboxyl group with the negatively charged oxygen, and we have lysine, which has that NH3 plus group at the very end. Now if these two amino acid residues are facing one another, they are able to interact favorably through electrostatic interactions. And now we can also understand that if we had two negatively charged R groups, so if we had a glutamate and an aspartate, and they both have negatively charged R groups at very high pH values, they're going to repel one another. So this can actually be a disruptive feature as well for tertiary structures. When we have opposite charges, they will attract and this will be favorable, but when we have like charges, they will repel one another and this will be disruptive to the tertiary structure. So we've already discussed two factors that could disrupt tertiary structure. It's when the pH reduces below the pKa of cysteine and we no longer have this disulfide bond. That would disrupt the structure. We would also disrupt the structure when we have like charges that are repelling one another and creating that unfavorable interactions, which will once again disrupt our structure. Now, thirdly, hydrogen bonding. We've seen hydrogen bonding in secondary structures on the backbone of our peptides, but here we are going to see it in the backbone of our peptide and on our R groups. Our R groups that have hydrogens and negative or positive charges are now able to interact via hydrogen bonding. Lastly, we can have metals. We can have any cations that have a +2 charge. So an example could be zinc. Zinc or magnesium. These are two very common atoms that are used in biological processes to balance out charges. So, for example, we could have, once again, our glutamate, or we could even have aspartate, and we know that it has a negative charge. That negative charge could be balanced out by a metal with a +2 charge, because remember that this oxygen has an extra 2 electrons, so it would need a metal that has a +2 charge to cancel out the 2 electrons that have a negative charge. So overall, we've seen that tertiary structure, the main driving force is the hydrophobic effect, where the nonpolar hydrophobic R groups cluster in the center and we get the hydrophilic molecules on the outside. Within tertiary structures, we do see beta plated sheets. We see the alpha helix. We could have disulfide bonds. And we could have disruptive forces, including no longer having disulfide bonds. We could have electrostatic repulsion. We could even lose hydrogen bonding, because if we increase our pH significantly, our R groups, this is going to be very important, our R groups, they will lose their hydrogen, so they will no longer be able to interact via hydrogen bonding. But remember that our peptide bonds, they will still retain their hydrogens. So the secondary structure actually will not change, because they do not have pK values. They are not ionizable, our peptide bonds. So they will still interact via hydrogen bonding. But the R groups, they will lose their hydrogens. The R groups that are ionizable will lose their hydrogens, and they will no longer be able to interact via hydrogen bonding. So this is a very important example where we can understand that if we are changing the pH, we will affect the tertiary structure significantly, because we're affecting the protonation state. But the secondary structure will remain same, because in our secondary structure, our groups are not interacting. It's our backbone that's interacting, and the backbone is not ionizable. And lastly, we can also have metals to balance out the charges. And that is essentially our tertiary structure. It is when the distinctive feature of tertiary structure is when our R groups are able to interact.